Um, I find that glaucoma talks get very repetitive, and I'm the sort of glaucoma doctor who's interested in the neuroscience of it more than the plumbing of it. And so my talk is really on some topics that I'm interested in, but it might be quite sort of heavy on the science, and it might not be everybody's cup of tea. So I will take no offense if anybody wants to just leave because it's all a bit nerdy. Um, but if anyone's interested and has questions, I would love to hear them as well. So I wanted to talk about a few things that are of interest to me, which is a bit bold of me because one of the things I want to talk about are my own projects. The first is about the genetics and where we're at with genetic testing for glaucoma. And then the second thing is about nicotinamide, which is a very trendy topic in glaucoma research, and lots of people are already recommending nicotinamide, which is vitamin B3 for their patients. And then the third thing is about the way in which our eyes are deformable and respond to pressure stress. And I've been having fun doing some projects tipping people upside down and considering the pressures not just in their eye, but in their head. So I'll enjoy telling you about those things. So there have been some really big developments in glaucoma genetics just in the last few years. So for years, we were told that glaucoma is mildly, runs in families to some extent. A twin study is a very powerful technique for measuring how much of a disease is due to genetic problems or genetic variation. And that suggested that only 13% of glaucoma was due to genetics. But people have considered that to be an underestimate. And in general, the advice has been that if you have one person in your family who's got glaucoma, one of your primary relatives, so your parents or your siblings, then your risk over your lifetime is 25%. And yet the only gene we had, and our most potent glaucoma gene still, is myocillin, and it only explains 1 or 2% of glaucoma. So there was a big gap, and we were looking for the genes that explained the big gap in between what we know about family history and the only genes we had found. And so this was a study from just a few years ago that I just thought was such a cool idea. So there are these huge hospital networks in New York City, and you can see that together these three big hospital networks had about 2 million people in them. And all of these big hospitals in New York are run on electronic records. And so you can mine those electronic records and preserve people's privacy and dig out lots of details about them without kind of betraying their privacy. And they went into the emergency contact details. If you have a record at the hospital, it will probably say, in an emergency, call my daughter. This is her name and her address. And if there's an emergency, call my husband or my father. And those kind of relationships could be built up from the emergency contacts to make, 600,000 different family trees with these people who've been looked after in these hospital networks. And these are some examples of the family trees that were kind of generated by mining the electronic record. And then you can label each of the people in the family trees with all the different diseases that they've been seen in the hospital for. And so I think that's just such an amazing, clever use of data. And the takeaway message, the thing that was most interesting to me was that out of all the very many conditions that they looked at, glaucoma was one of the most inherited conditions. And the great majority of glaucoma can be explained by genetic variation. And so this is, this sort of is comforting to those of us who thought that there was quite a lot of genes involved. And um, the status of glaucoma genetics is more about looking at multiple weak genes rather than one or two genes that explain the condition. And so a genome-wide association study is where you look at these variable sites in the genome. So our genome is thought of as like an encyclopedia with 22 volumes. And in each one, there are millions of letters forming genes in a big recipe book for how to make each individual. And some of the letters in the encyclopedia are single nucleotide polymorphisms, meaning they're places in the genome where there's at least 5 or 10% of the population have a different letter in that location. And so those points that are variable, they don't really tell you anything about the gene in that location. They're just variables that tend to go together, and they tell you about a location in the genome. And so you can do these these genome-wide association studies looking at millions of these variable sites, and you get a plot like this called a Manhattan plot. And that's where areas that are associated with glaucoma sort of pop out of the general noise and show that there's probably something in that location which is associated with glaucoma. And our main glaucoma gene, myocilin, is this one over here. And so this has revealed multiple places where there are genes that are linked to glaucoma. But each of the SNPs themselves is not a glaucoma gene. It's a variable site that indicates there's something in the genome around there that is relevant to glaucoma. But all of these variations are present in normal people and present in people with glaucoma, and each of them is just associated with a slight upward or downward change to your glaucoma risk. So if the blue people here have got glaucoma and the people colored white are people without glaucoma, then you could imagine that the green SNP was slightly protective against glaucoma. Maybe the orange SNP is slightly irrelevant to your risk of glaucoma because it's the same amount in the white and the blue people. Anyway, the SNPs are present all through the population, and each of them is just associated with glaucoma rather than being a proper glaucoma gene. And so we're now up to about 100, maybe 150 locations in the genome that are related to glaucoma. Not really glaucoma genes, but somehow associated with glaucoma. And some of those places in the genome you could imagine would be associated with having a high pressure, or with being short-sighted, or with a specific type of glaucoma like angle closure or pseudo-exfoliation glaucoma. And we've sort of got the impression that we're heading to about 200 glaucoma locations, or 200 glaucoma genes. Will probably be about the number that we get when we've done as many of these studies on as many tens of thousands of people as can be achieved. And so this result, just to cut a complicated story short, indicates that about 75% of glaucoma is genetic, based on the 140 genes that are being tested in these polygenic risk scores, these SNP analyses. And that kind of fits with the New York electronic record study. It indicates that glaucoma is very heritable, but it's inherited from many very weak genes that don't contribute much. So you can now get a polygenic risk score. You can pay for one from a specialist glaucoma lab, or you can just go on 23andMe and send your DNA to the States, and they'll send you back lots of information about your heritage, but also about your risk of various common chronic diseases. So you might get a risk score for diabetes or heart disease, and a lot of these products are offering a risk score for glaucoma. And achieving that and recognizing that we can estimate genetic risk doesn't necessarily answer the question about what we should do with this. Genetic testing always has a few unexpected risks involved, because you're not just finding out information about yourself or about your patient, you're finding out information about other people in their family who've not really asked for that information. And so there are important considerations about the way in which finding out that information about one person could affect other people. For example, finding out that your lifetime risk of Huntington's disease, which is a horrible type of dementia that strikes in middle age and is very disabling and leads to an early demise. Huntington's disease is a terrifying genetic diagnosis to have, and so a lot of people choose not to have that information. And so in a milder way, finding out that your risk of glaucoma is very high might sort of have a negative influence on your life rather than a beneficial influence on your life. And I mean, it hasn't been a big problem in New Zealand that people are denied health insurance because of genetic information. But those kind of considerations are out there. So I guess we need to be cautious about genetic testing, especially as we roll it out into bigger and bigger groups of people. And these risk scores, although they tell us a lot about glaucoma risk, they're not very useful in terms of predicting glaucoma on a day-to-day basis, because they're about your lifetime risk. So if you test a whole lot of people in their 30s, even the people with a high genetic risk will probably not have glaucoma. And so it's been estimated that you have to do hundreds of tests to find a single person who's got glaucoma at that time. And so that doesn't make it very good for screening the population. So I think there are a couple of places where these risk scores might be really helpful, and that is for people who have a strong family history of glaucoma. So if your sister has glaucoma, you might be interested to know whether you're at high risk or normal risk. Do you have the same genes that she has that have contributed to her glaucoma, or are you at a lower risk and don't need to go to the optometrist so much for screening? And then the other group are people who've got some glaucoma, but not terrible glaucoma. And it might help us to modify the follow-up plan for them. So if they've got some glaucoma, but they seem to have a favorable genetic profile, then they might just need a checkup once a year. And if they've got early glaucoma and an unfavorable genetic profile, then they might need to continue regular follow-ups forevermore. So there are a couple of groups where genetic risk scores might be useful, but all of these ideas sort of need to be proven by proper clinical studies, where they have a large group of people and only do genetic testing on half of them to decide whether finding out that information has a beneficial effect on their long-term outcomes. So that was all I was going to say about genetics. The second thing I wanted to talk about was nicotinamide. And this is vitamin B3, and the widely available brand in New Zealand and Australia is the Blackmores and Solar product. I am in no way promoting this product. I'm not, I don't have any conflicts of interest. It's just, this is the vitamin B3 product that's out there. And this is really a story of two interesting little black mice. There is a type of mice called the DBA2J mouse, which has a type of pigmentary glaucoma. And there's another type of little black mouse called the Wallerian degeneration slow mouse. And together these have led to the understanding about nicotinamide. And I think it's an interesting sort of chronological story about how nicotinamide has become such a trendy idea for glaucoma. So this is a cartoon of the back of the eye, and the optic nerve cells, the retinal ganglion cells, they live in the retina, and they're dotted all over the retina, and they receive visual information from the photoreceptors. And then they send their nerves across the back of the eye and out to the brain. And this part further back from the eye has got myelin around it, and that makes this part of the nerve relatively robust. So the bit behind the optic disc is relatively strong, and we can tell from the pattern of visual field loss and how it gets worse over time that when ganglion cells are damaged and die from glaucoma, the damage is occurring at the optic disc. And when a cell is damaged on its axon, on its nerve fiber, the first thing that happens is that the downstream piece of the axon is disassembled. It's broken up into pieces and absorbed by other cells. And that's a sort of organized type of disassembly called Wallerian degeneration. And it's one of the earliest things that happens to a neuron when you stress the axon. And then there is transmission of sort of unfavorable toxic signals to the nerve cell body. And this part of the axon subsequently degenerates. And then the cell undergoes a sort of non-inflammatory, gradual, controlled process of cell death called apoptosis. So when we're looking at things that will help glaucoma, we want to look at the early processes. There's no point saving a cell that's on death's door and doesn't have any connection to the brain anymore. So it's these early signals and early pathways that are useful in protecting cells against glaucoma. And so the DBA2J mouse was a relatively new model. It's only been around for 5 or 10 years. And it's not a perfect model. It doesn't behave the way that human glaucoma does. It's a relatively aggressive type of glaucoma. The mouse obviously doesn't live as long as a person does, but the mouse undergoes pretty devastating glaucoma in its first year of life and gets cataracts and other problems in the eye that makes it a bit different to human glaucoma. But what they did was they looked at the mouse at 4, 5, 6 months of age as glaucoma was just starting to become developed, and they took the ganglion cells out and looked at which pathways in their genetics had been modified compared to the younger, healthier mice or to ones who didn't have the glaucoma mutation. And what they found was that the changes in the ganglion cells were metabolic genes leading to energy production and metabolism of fuels and oxygen, in particular the mitochondrial energy production pathways. And in particular, this enzyme called NMNAT2 was reduced in activity in the damaged cells, which led to a deficiency of this NAD molecule. And so it appeared that in early glaucoma, a deficiency of NAD was a vulnerability factor, and replacing the NAD protected the cells against death. And in the other mice, it was interesting to study this mutation and realize that what it did was transport NMNAT from the cell body out to the axon where it was helpful. And that supported levels of NAD in the axon. And that's why these animals did not undergo Wallerian degeneration in glaucoma or other axon injuries. So these two mice kind of really highlighted how this enzyme called NMNAT2, which produces NAD, is really important in early glaucoma, and how a loss of NAD plus is important in making the cells susceptible to damage and death from glaucoma. And so NAM is nicotinamide, and that's the substrate that is turned into NAD, and that's vitamin B3. And so those observations in the little black mice led to this work, mostly led by Pete Williams here. And you'll see he's got his fingers in nearly all of this work. It's really turned his career. He's now like a superstar. This is a big paper in one of the biggest science journals in the world. And this has really launched him into scientific stratosphere. And what they found was that in the control mice, the retina has got a thick layer of ganglion cells and the optic nerve is full of nerve fibers. In the eyes with the DBA2J glaucoma, they had fewer ganglion cells and fewer nerve fibers. And if you gave them nicotinamide, you protected them against loss of ganglion cells and loss of nerve fibers. And it was really quite an impressive paper. And they drew a nice picture like this that shows that the NAD deficiency contributes to multiple types of degeneration. Multiple aspects of ganglion cell degeneration are affected by NAD deficiency, and that supplementing it through nicotinamide tablets prevented all these different aspects of ganglion cell degeneration. So it's not just about protecting the axon, it's about protecting the dendrites, it's about preventing unhealthy gene expression changes, loss of the little connections called synapses at both ends of the cell. So nicotinamide has become super trendy and super popular because it does seem to be a wonder drug for ganglion cells in glaucoma. And so the mice have launched a whole lot of investigation into people with glaucoma. So this was a study done in Melbourne by some of my favorite colleagues and doctors, and you can see Pete Williams is in the mix there again. And this was using electrophysiology, these signals from the surface of the eye, as an indicator of whether the ganglion cells were under stress or not. And they took people with glaucoma who seemed to be getting worse, people whose target pressure was, their eye pressure was not controlled, or they were getting worse on the visual field test, and they randomized them to have nicotinamide tablets or placebo tablets and measured the electrophysiology after 6 weeks and 12 weeks to see whether the measures of cell stress improved. And the answer is they did. But measures of cell stress is one thing. It's cell survival and visual fields over time that are important to doctors and patients. So this is a study from New York where they took only about 30 people. There we go, 32 people. And they randomized them to have nicotinamide or placebo. The 21 with nicotinamide were not the 21 women. They were randomly analyzed, randomly assigned. And then they measured their visual fields after 7 or 8 weeks. And the people in the taking nicotinamide had improved visual fields, and more of the places in their visual field were improved. And this was said to be a positive, statistically significant result. But when Pete Williams has told me, he said things like, "Are you taking nicotinamide? It's very nice to take it. It's like taking lots of coffee." And that makes me think that it would probably make me do a better job on a visual field, even if my optic nerve hadn't changed at all. So this result doesn't really impress me either, because if you gave people a few cups of coffee and then asked them to do more visual field tests, they would probably do a bit better. And it's really the long-term visual field function and survival of the ganglion cells that will determine whether this is an important treatment or not. So there are multiple studies around the world ongoing that are trialing whether nicotinamide has a meaningful long-term effect in people. And there are also people using creative approaches to boost NAD in ganglion cells using gene therapy or using molecules from green tea, which are under investigation. because they increase the activity of that enzyme and increase production of NAD in the ganglion cells. And they might be more effective or more have a better kind of side effect profile compared to just taking lots of nicotinamide. All right, so that was the nicotinamide story. And then the last thing I wanted to talk about was the sort of biomechanical side of the eye. This is the way in which the eye pressure damages the optic nerve, and a couple of projects that I'm enjoying of my own. So our head has got a few pressurized compartments, the eye pressure that we're all quite familiar with in the glaucoma clinic, and then the intracranial pressure, which tends to be a bit lower and is usually measured when you're lying down by a lumbar puncture, which is quite an invasive measure. And when we stand up, the intracranial pressure probably drops even further, so it's pretty close to zero when we're standing up. And so this idea that there's an eye pressure and a head pressure, which is pretty equivalent to the optic nerve pressure, these pressures are in opposition at the optic disc, and that creates a force across the optic disc, which tends to push backwards because the intraocular pressure tends to be higher than the optic nerve. And that pressure is called a translaminar pressure, meaning across the lamina, which is the colander structure at the optic disc. And when the eye pressure is high over years, the optic disc is pushed back, and that is our understanding of what's happening in glaucoma. But we know that the pressure of the eye on its own is a very poor predictor of how much glaucoma someone has. This eye has got a healthy optic nerve, but a high pressure in the eye. And this eye has got a low pressure in the eye, but a fragile, progressive glaucoma problem. And so we sort of understand that there's variation between people. Some people have a rigid eye like a ping pong ball, and some people have a stretchy eye like a balloon. And here's an example of a remarkable eye that's very much like a balloon. When the pressure is high, the optic nerve is large and it has a big cup in the middle that looks a bit like glaucoma. And when they have glaucoma surgery, the optic nerve shrinks, the pressure is low, and the big cup goes away. And then when the surgery stops working, the pressure goes up and the cup gets bigger. And when they drop the pressure, so you can see there's an extremely elastic eye, and it remains an area of debate as to whether having a stretchy eye is a helpful thing or a harmful thing in glaucoma. It's really not clear. And the reason it's not clear is that you've got the translaminar stress that I talked about, but you've also got this hoop stress, like the example I just showed, where the whole eye is elastic. And if the wall of the eye is elastic, then it tends to reduce translaminar stress on the optic disc. Whereas if the wall of the eye is rigid, then any variation in the pressure is concentrated at the optic disc. And so trying to get our head around these two different types of stress on the eye and on the optic nerve is a very complicated project. So one little contribution that we've been enjoying in Wellington is tipping people upside down. And the major achievement of this work was coming up with a funny acronym. So the idea was we were looking at the pulsations in the retinal vein because they indicate the translaminar pressure. And we've called it the Space Force study because it's sort of relevant to the eye problems that astronauts get, but I didn't get any money from the Space Force or from the United Federation of Planets. So the main innovation was to put the tipping upside down table up on some pallets so that when you're upside down, your head is not down here by the floor, but more than a meter off the ground so that you can do an OCT scan. And that's allowed us to do all sorts of interesting measurements of the eye when people are upside down. And the first set of measurements are just about the eye pressure. So this is the intraocular pressure. And here's someone sitting up. and the pressure was about 14. And as you tip your head back and tip the table back, the pressure doesn't change much. And when you lie down flat, it's only gone up to about 17. So it hasn't changed much. But then when you go from lying flat on your back to having head down and then being totally upside down, the eye pressure behaves like a fluid column, very much behaves like a passive physical process. The pressure goes up immediately within 1 or 2 seconds of changing the angle, and it stays there while you're in that angle. And so this indicates that the eye pressure with head down is driven by a fluid column of the veins from your eye to your heart. The length of the fluid column is about the distance from your eye to your heart. So that's interesting. So when you tip upside down, everyone knew that the eye pressure goes up, and everyone knew that the head pressure went up. But the part that's new is understanding whether the eye pressure goes up more than the head pressure, or whether the head pressure goes up more than the eye pressure. And so these are little videos of somebody's optic disc when they're sitting up, and the same person when they're upside down. And you can see that this vein, this retinal vein, is pulsing, and then it's still pulsing when they're upside down. And it's pulsing by a similar amount, even though their eye pressure has gone from 20 to 40. Now, if someone was sitting at the microscope and we squeezed their eye so that the pressure went up to 40, the pulse at the vein would get a lot bigger. So the fact that the pulsation didn't change much indicates that their head pressure went up just as much as their eye pressure. Here's another person. It's a little bit of a pulse when they're sitting up, and there's a little bit of a pulse when they're upside down. And so even though their eye pressure has gone up a lot, their head pressure has gone up just as much. And so this is just a grading on this axis, a grading of how much pulsation there was. And most people, the lines are horizontal, meaning there wasn't much change. Now, there's a few people where the pulsations got less, and one person where both eyes showed more pulsation. So this is somebody who was sitting up and they've got a decent pulse in this vein. And when they go upside down, that pulsation has diminished. And that indicates that their head pressure has gone up more than their eye pressure. So that's interesting. What's different about this person compared to the others? And then here's the opposite. The person has a mild pulsation when they're sitting, and a very much greater and more obvious pulsation when they're upside down. So that indicates that this increase in their eye pressure was greater than the increase in their head pressure. So again, that's interesting. Why is this person different to the others? Is there something about their anatomy or their veins that stops their head pressure going as much? So anyway, there's lots more to think about there, and I will start recruiting some of my friends who have glaucoma, or my patients, I guess, to put some more people with glaucoma on the upside down table to see whether their responses are the same or different to people who don't have glaucoma. I've had 2 of my generous patients offer to go upside down. And the reason that they were keen to do that was that they've got a sore back and they wanted to stretch their back on the tippy table. That's why you can buy them at the sports shop. And they wanted to know whether tipping upside down for their back pain was going to be damaging to their glaucoma. And the takeaway message was that in both of those people with glaucoma, the pulsations did not change. So just like other healthy people without glaucoma, their head pressure went up at least as much as their eye pressure did. And so even though the eye pressure goes up a lot, the pressure behind the optic nerve goes up to a similar extent. And so the amount of stress on the optic nerve is much less than if you just increase the eye pressure. And that's a new interesting model of glaucoma. So it might change how we advise people who want to do yoga and various other activities that tip their heads down. And there might be some people who would benefit from a wedge under the mattress when they're asleep at night, so that their head is up a little bit or down a little bit, depending on what the pulses of the vein suggest. It's still definitely an area of active inquiry. I don't have any recommendations for glaucoma patients at this point. And then the other last project that I wanted to tell you about was a collaboration with John, who's from South Dakota, and with Sam Holdsworth, who's in Gisborne. And John is a glaucoma doctor. He has developed these goggles. He had an idea, which I think might not be correct, that putting a vacuum over the whole orbit expands the eye, lowers the pressure, and that might be helpful for glaucoma, just putting a bit of a vacuum suction over your eyes for half an hour or an hour or even overnight might be beneficial for your glaucoma. I don't know if I agree with the reasoning there, but he made a custom pair for us where we could apply a positive or a negative pressure for one eye or the other. And so we've been doing that at Matai Medical Research Facility, which is in Gisborne, and it's led by this very remarkable, impressive person, Sam Holdsworth. She is a MRI genius. She's kind of a physicist who can drive an MRI machine the way that an eye doctor can drive a slit lamp microscope. She can just tweak all the knobs and buttons, and she's constantly moving and talking and thinking, and she can generate new signals from the MRI, which is really a great privilege to be able to do kind of projects with someone like that. And so you can see here's somebody in the MRI scanner with the goggles on, and this one's under positive pressure and it's kind of puffed up and pushed off the face, and this one's under negative pressure and it's kind of sucked onto the face. And so we have been putting people in the scan and applying negative and positive pressures to the whole orbit. And what we're looking for here is which parts of the orbit, meaning the eye, the optic nerve sheath with the spinal fluid around it, the fat and the muscles, which parts of the orbit are most deformable and changeable when you apply a pressure, and which parts are more rigid and resilient. And we're trying to get a sense of the deformability and displacements of different structures within the orbit, not just at the optic disc, but all the broader aspects of the eye. The other thing that Sam has developed that is quite innovative and interesting is to amplify the little movements in the orbit. So when you record an MRI at different parts of the cardiac cycle, different parts of the heartbeat, there are these little pulses that occur just from the filling and emptying of blood vessels and the variation in blood pressure throughout the systole and diastole, the cardiac cycle. And usually those movements are too small to see. And so Sam has developed mathematical image processing approaches that amplify those signals and make the eyes move, make the structures on the MRI scan move more than they truly are, but in the same direction that they truly are, so that you can see them with the naked eye. And so these wriggly-jiggly movements have never been observed before. They're a new finding, and we really don't know if they're important or completely irrelevant. But it's fascinating to try and develop a vocabulary and work out what's normal, what's not, and is it changing when we apply pressures. So we've got an ongoing project with Sam in Gisborne working on that. So anyway, thank you all for bearing with me to the end. Do we still have anyone in the audience? I appreciate that this is pretty dry and academic stuff, but I hope there was something of interest in there for you. And if anybody has questions, I'd be really thrilled to discuss these things more because they're right up my alley.